Part 2 of this cancer lecture. So, oncogenes, as described earlier, oncogenes encode proteins that promote the loss of growth control and the conversion of a cell to a malignant state. Oncogenes are derived from proto-oncogenes, which are genes that encode proteins having a function in the normal cell. Numerous oncogenes were initially identified as part of the genomes of RNA tumor viruses, but many more have been identified because of their importance in tumorigenesis as determined in laboratory animals or human tumor samples. Different oncogenes become activated in different types of tumors, which reflects variations in the signaling pathways that operate in diverse cell types. The oncogene mutated more frequently in human tumors is RAS, which encode a GTP binding protein RAS that functions as an on-off switch for a number of key signaling pathways controlling cell proliferation and metabolism. Oncogenic RAS mutants typically encode a protein whose GTP's activity cannot be stimulated, which leaves the molecule in an active GTP bound form, sending continuous proliferation signals along the pathway. Despite extensive effort to develop anti-RAS strategies for cancer therapy, no drug that block RAS function have yet to be approved. The function of a number of oncogenes are summarized. Oncogenes that encode growth factors or their receptors. Oncogenes that encode their growth factors or their receptors. The first connection between oncogene and growth factor was made in 1983, which was discovered that the cancer-causing simian sarcoma virus obtained an oncogene derived from the cellular gene for platelet-derived growth factor (PDGF), a protein present in human blood. Cultured cells that are transformed in this virus secrete large amounts of PDGF into the medium, which causes the cells to proliferate in an uncontrolled fashion. Overexpression of PDGF has been implicated in the development of brain tumors. Overexpression of PDGF has been implicated in the development of brain tumors or gliomas. Another oncogenic virus, avian erythroblastosis virus, was found to carry an oncogene ERBB that encode an EGF receptor that is missing part of the extracellular domain of the protein that binds the growth factor. One might expect that the altered receptor would be unable to signal a cell to divide, but just the reverse is true. This altered version of the receptor stimulates the cell constitutively, that is, regardless of whether or not the growth factor is present in the medium. This is the reason why cultured cells can carry the altered gene proliferate in an uncontrolled manner. A number of spontaneous human cancers have been found to contain cells with genetic alterations that affect growth factor receptors, including EGFR. Most commonly, the malignant cells contain a much larger number of receptors in their plasma membranes than do normal cells. The presence of excess receptors makes the cell sensitive to much lower concentrations of the growth factor, and thus they are stimulated to divide under conditions that would not affect normal cells. Growth factor receptors have become a favored target for the therapeutic antibodies, which bind to the receptor's extracellular domain, and for small molecule inhibitors, which bind to the receptor's intracellular tyrosine kinase domain. An oncogene that encodes cytoplasmic protein kinase. Overactive protein kinase function as oncogenes by generating signals that lead to inappropriate cell proliferation or survival. RAF, for example, is a serine threonine protein kinase that heads the MAP kinase cascade, the primary growth controlling signaling pathway in cells. It is evident that RAF is well positioned to wreak havoc within a cell should the enzymatic activity become altered as the result of mutation. As with the growth factor receptors and RAS, mutations that turn RAF into an enzyme that remains in the on position are most likely to convert the proto-oncogene into an oncogene and contribute to the cell's loss of growth control. RAF is most closely linked to melanoma, where BRAF mutations play a causative role in the development of approximately 70% of these cancers. Another group of cytoplasmic kinases that are often deregulated in cancer are the cyclin-dependent kinases. Cyclin-dependent kinases, especially CDK4 and CDK6. Cyclin D1, a regulator of these CDKs, is also a frequent oncogene. And the first oncogene to be discussed and discovered is RC. Is also a protein kinase, but one that phosphorylates tyrosine residues on protein substrates rather than serine and threonine residues. Transformation of a cell by SRC containing tumor virus is accompanied by the phosphorylation of a wide variety of proteins. Included among the parent substances SRC are proteins involved in a signal transduction, control of the cytoskeleton, and cell adhesion. For the unknown reason, SRC mutations appear only rarely among the repertoire of genetic changes in human tumor cells. And oncogenes that encode transcription factors. A number of oncogenes encode proteins that act as transcription factors. The progression of cells through the cell cycle require the timely activation or repression of a large variety of cells whose products contribute in various ways to cell growth and division. It is not surprising, therefore, that alterations in the protein that control the expression of these genes would seriously disturb a cell's normal growth patterns. Probably the best studied oncogene whose product acts as a transcription factor is MYC. MYC regulates the expression of a large number of proteins and non-coding RNAs like rRNAs, tRNAs, and miRNA involved in cell growth and proliferation. When an MYC expression is selectively blocked, the progression of cell through G1 is blocked. The MYC gene is one of the proto-oncogene most commonly altered in human cancers, often being amplified within the genome or rearranged as the result of a chromosome translocation. These chromosomal changes are thought to remove the MYC gene from its normal regulation influences and increase its level of expression in the cell, producing an excess of MYC protein. One of the most common types of cancer among populations is in Africa called Burkitt's lymphoma, results from the translocation of MYC gene to the position adjacent to an antibody gene. The disease occurred primarily in persons who have also been infected with Epstein-Barr virus. This same virus causes only minor infections, mononucleosis, in people living in Western countries, as it is not associated with tumorigenesis.